Let us do Brahmanand.
Hello students. In the previous sessions of the biotechnology, we discussed the tools of biotechnology, the steps of rDNA technology, the rDNA recombinant DNA technology. In recombinant DNA technology, we discussed the steps starting from the isolation of the passenger and vector DNA, cleaving of passenger and vector DNA by using the same restriction enzyme, restriction endonuclease, then the separation of the fragments of DNA fragments, fragments of passenger DNA by using the gel electrophoresis. Then then separated fragments, they are transferred to the nylon membrane, that is nitrocellulose membrane, by blotting method, that is southern blotting for DNA separation for the transfer of the DNA fragments. Then the recombinant DNA is produced, which is called chimera. Then they are introduced into the host cell for cloning, or they are cloned by using the PCR, polymerase chain reaction. The cloning can be done by host cell based or the polymerase chain reaction based, we discussed. And before introducing them into a host cell or we have to select. So we have selected that there is a selection procedure, that is through the insertional inactivation. The transformed cells are then, after introducing into the host cell or into the target cell for expression, the transformed cells are selected, screened and selected by the process of by using the selective markers and the chromogenic marker by the process of insertional inactivation. So what we learned? We learned the steps of rDNA technology, how to develop the recombinant DNA. Now in this session, we are going to discuss the application of the rDNA, recombinant DNA, for the development of the products for the mankind. So what is biotechnology? As we discussed, it is an integrated and applied branch of biology which deals with the use of microorganisms or plant cells, animal cell or fungal cell, or the whole organism for the expression of the desired products and services for the mankind. So either the cell microbes or the cells of the fungi, plantae or animalia, or the whole organism is modified by using the genetic engineering by the process of gene manipulation. The genetically modified organisms are developed for the production of the useful substances and services for the human being. And this biotechnology is used mainly for the production of food and health. So we can say the application is done for the in agriculture and the health. According to the use of the biotechnology, on the basis of the use of the biotechnology, it can be of 4 types. It can be red biotechnology, green biotechnology, white or gray biotechnology, and blue biotechnology. Okay, so write down the definition. Biotechnology is an integrated and applied branch of science which deals with the use of microbes, plant cells or animal cells or the whole organism for the formation of products, beneficial products and services for the mankind. GMOs, the genetically modified organisms. Here we have discussed that genetically modified organisms. Okay, genetically modified organisms have been altered by gene manipulation, by altering the gene, and application ranges from the agriculture to health and industries to environment. According to the application of biotechnology, it is a 4 types. Green biotechnology. Green biotechnology is used in the field of agriculture. In the field of agriculture for crop improvement. For crop improvement. Red biotechnology. It is the use in the field of or application in the field of medicine. Field of medicine. Green white biotechnology. It is in the field of industries to develop a large scale products for the development of the products, beneficial products in large scale in the field of industries. Blue biotechnology, it is in the field of marine field for we can say sea products, marine products or for sea products we can say. Okay, so sea for the sea organisms, sea products, sea organisms, marine organisms you can say, that are having the food and medicinal value. So let us discuss in different fields. The main fields that are used, applied, biotechnology applied is agriculture, and that means application in plants, application in medicine, and it is made beneficial for the animals or animal products for the mankind. So application in plants, application in medicine and application in animals, mainly in these three areas we will discuss. So let us discuss the application in plants. Plant cell and tissue culture. By adopting the plant genetic engineering, the plant cell genome is altered. Alteration of plant cell genome is done by transferring the desired gene of one plant cell to another plant cell. So what is done, and then it is cultured. Then the transformed cell is cultured to develop the tissue, to develop the particular that multiple copies of that plant cell, then from which the tissue and then the plantlet, then plant can be developed. So the product can be obtained. So what is done? Plant cell and tissue culture. How it is done? The plant genetic engineering, by using the plant genetic engineering, alters the plant genome of one cell by transferring from one cell to another, by transferring the desired gene from one cell to another. Then the transformed cell is cultured. Transformed cell is cultured in the optimized condition by the process of genetic engineering. It is cultured in the laboratory in the optimized condition. What factors are required in that condition? The cells are cultured to develop into plantlet. So transformed cells cultured in the optimized condition to develop the clones of that cell, then tissue, then plantlet, then plants. So obtaining the products. Now these plants are having the recombinant gene. The DNA is recombinant type. Okay. So similarly, the drought resistant plants, the drought resistance which can withstand in the lake of water in the drought conditions, the plants which can withstand for the longer time in the drought conditions. So which plants do the less transpiration, which hold the water for a longer period. The genetic engineers, they already find out the drought resistant plants, and the genes of that drought resistant plant are transferred to the developing crops which are required for the human beneficial crops. So drought resistant, the genes are transferred. The genes are introduced into the crops. Which genes? The drought resistant gene, the genes of the drought resistant plants. The genetic engineers identified the drought resistant plants, genes are introduced into the crops to enhance the water holding capacity, to enhance the water holding capacity, so that they can withstand in the drought condition. Fungi, bacterial resistant plants. Some plants are having the inherited that characteristic that they can resist from the fungal or bacterial infection. So those the genes from those plants, bacterial or fungal resistant plants, are introduced into the into our desired target crops to make them free from fungal and bacterial diseases. Okay, gene from fungal and bacterial resistant plants. Gene from fungal and bacterial resistant plants are introduced into our desired crops, are used, introduced into the target crops. The virus resistant plants, the gene that encoding the coat protein, the capsid that is the coating over the genetic material of the virus, the gene encoding for the coat protein are introduced into the cells, into the plant cells for the development of that coat protein, so as the plant immune system is developed. Plant immune system is developed. The gene encoding for coat protein of infectious viruses, of infectious plant infectious viruses, of plant infectious viruses are engineered, are introduced by genetic engineering, are introduced or recombined with the plant's genome. In the target's plant genome, introduced into target crops to by genetic engineering, so as to by synthesizing the coat protein by that crop plants, the immune system is developed. The plant's immune system is developed, protecting itself from the viral infection. So what happens? By introducing this into the target crop, the coat proteins are synthesized. Coat proteins are synthesized so as developing the plant immune system. Plant immune system is developed. Thus, the plant protect itself from the viral infection. So, plant protects itself from the viral infection. Is that clear? Then insect resistant insect resistant or pest resistant plants are developed by synthesizing the pest resistant protein. How? Some bacteria, they produce some that protein which can resistant against the pests. Means some bacteria, they synthesize the pest resistant protein, and if that protein can be synthesized by the plant itself, they can be protected from the pests, especially from the insects. We will discuss this in BT crops. We will discuss in details in BT crops. What is BT crop? That Bacillus thuringiensis, the gene from Bacillus thuringiensis. Write down all this. The gene encoding for Bt protein from Bacillus thuringiensis is incorporated in the plant genome for the synthesis of Bt protein. This Bt protein is capable to destroy the pest, especially the insects resistant to pest, or especially the pest means who gives harm to another. So the insects, mainly insects or pest resistant plants, are developed by producing the Bt proteins. How it can be? Through genetic engineering, the gene encoding for the Bt protein from the Bacillus thuringiensis is transferred into the plant cell. Then plants are this type of the Bt crop plants are developed by the process, by using the genetic engineering. Herbicide resistant plant, the bacterial herbicide resistant gene is transferred to the plants. Bacterial resistant genes are transferred to of course target plants. In which plant we want to have this character, we want to express this character. So bacterial resistant genes, bacterial herbicidal resistant genes, some bacteria, they are resistant to herbicides. They are sprayed on the crop plants to kill the wild herbs which are not having any economic importance. Herbicidal resistant genes are genetically transferred, or we can say incorporated in the crop plants to make them herbicide resistant into the target crop plant. How? In the plant cell. They are into the target plant cell. Then from this target plant cell, they are cultured. They are cultured to develop the herbicidal resistant plants. To develop the herbicide. Okay. These arrows I have given due to the lack of space. You can give downward arrow. You can use the downward arrow to write down the points. Frost resistant plants. What happens to the plants when the frost is formed? Okay, frost causes the formation of ice crystals. Uh, Ice crystals are formed. Frost causes ice crystals to be formed in plant tissue. So that leads to the abstraction of that destruct the fluid transport. The water content what is present within the tissue that gets freezed and becomes ice crystals to be formed in plant tissue. That means the moisture content, the water content present in the tissue becomes freezed and becomes the ice, and so the fluid movement is stopped. Fluid movement is stopped, is destructed. Then what happens? The leaves they exhibit water soaked. Leaves becomes water soaked. Leaves become water soaked in water. That results in wrinkle. Leaves become wrinkle and contract. That makes the water soaked and shrive. Shrive means wrinkle. Gets wrinkled. Leaves gets wrinkled and contracted. Leaves, what becomes contracted? Leaves get wrinkled and contracted, and thus that leaves and the plant tissue, even the plant tissue becomes dark brown and black. Leaves and plant tissue where the frost is formed, ice crystals are formed because of the frost. When you see in the in cold winter, water droplets found on the leaflet, on the leaves of the plants. Okay, that causes the crystal that ice crystal formation of ice crystal. So leaves and plant tissue becomes dark brown or black. So what will happen? No photosynthesis, chloroplast is destroyed. No photosynthesis. Plants die. So ultimately the plants die. Plants they cannot be stand without water circulation, so it causes the death of the plant. So how to get rid of this frost effect of the frost? A gene is present in the soil bacterium, gram-negative bacteria, Pseudomonas syringae. That gene is isolated and incorporated in the plant cell. In plant cell means in the callus, callus, C-A-L-L-U-S, callus, that is the undifferentiated cell. Into that we incorporate. Then from that GMO plantlet, then plants are developed. So in the cold region, the plants can be developed by incorporating the gene taken from the Pseudomonas syringae that causes the ice nucleation. that gene in the bacterium that causes the ice nucleation. That means it causes the formation of ice crystals on the membrane, on the cell wall. How? First of all, it synthesizes a protein and that are secreted onto the membrane, and that causes the formation of ice by taking the moisture of the environment. So, if this gene can be mutated, actually it is done by the genetic engineers, the scientists, already it is done. So, this gene is mutated, and this mutant variety is introduced into the plant cell. So, how it can be? By incorporating the ice minus, okay, from which bacterium it is taken. There is a soil bacterium, a soil bacterium gram negative, okay, it is gram negative bacterium called Pseudomonas syringae, Pseudomonas syringae. It is having the bacterium. These bacteria are having a strain. They are having a strain, that is gene. The strain of the gene is called the ice plus gene, or INA, the ice nucleation active protein that produces. So, the strain of Pseudomonas syringae, strain or gene that causes ice nucleation, and this ice nucleation gene is called ice plus or INA strain. INA strain. This INA strain, INA stands for INA. This INA stands for ice nucleation active. Ice nucleation active protein is produced from the ice INA strain. This gene, this strain causes the ice nucleation, is called and that synthesize a protein ice plus or INA strain. INA strain, INA strain, it's okay, that produces the ice nucleation active protein, INA protein, or ice plus protein, ice plus or ice nucleation active protein. What occurs? That causes the formation of a protein on the cell wall, that INA protein on the outer surface of bacteria. Outer surface of bacterium, that becomes the center of formation of ice crystal. That becomes the center of formation of ice crystal. So, thus we can say ice formation occurs. But if we will do the mutation of this, if the mutation is done to this strain. The mutation, if the mutant variety, mutant variety means that we can see INA minus strain. What we will tell this? INA minus strain. This is the, if mutation is done, the IS plus or the INA strain, then we can get INA minus strain. So INA minus strain, that will not form. IS minus strain lack the ability to produce. It lacks the ability to produce. It lacks the ability to produce the crystals, the crystal proteins produced to the to produce these crystal proteins are called surface proteins. Like the ability to produce surface proteins, that surface proteins which are formed on the membrane, on the cell membrane. Okay, so that it lacks the ability to produce the surface proteins, so no ice formation. No ice formation. So as the ice will not form, ice is not formed. So by introducing this gene, so what the genetic engineers do? The genetic engineers, they introduce GE means genetic engineering. Through a genetic engineering, this INA minus strain, is introduced into the plant callus. Then from callus, the plantlets are grown. Then GMO, frost resistant, GMO, genetically modified, GM, frost resistant plants are developed. Am I clear?